image like no other, inspiring decades of research and debate. The Shroud of Turin, that's a piece of history, and it has an ongoing puzzle for science.
The Shroud of Turin is a burial cloth that's imprinted with what many believe is the actual image of Jesus after his crucifixion. Now, it's been debated for years, but the shroud is back in the headlines in a major way.
Deep within the ancient fibers of the Shroud of Turin, scientists uncovered something astonishing. Human DNA. Advanced genetic analysis revealed mitochondrial DNA from multiple geographic regions, suggesting the cloth was handled by many people across centuries. For millions of believers worldwide, the shroud is believed to be the burial cloth of Jesus Christ, the only physical trace of the resurrection. A silent witness, a fifth gospel written not in ink, but in blood. But what exactly did this DNA reveal? And why did it leave researchers stunned? Was it evidence of ancient origins, or proof of something far more complicated? In this documentary, we break down the science, the controversy, and the discovery that has reignited one of history's greatest debates. If you're fascinated by mysteries, make sure to subscribe to the channel and turn on notifications, because the truth behind the world's most controversial relics is only just beginning to unfold. To skeptics, however, the shroud tells a very different story. They see it as the most sophisticated and convincing medieval forgery ever created. Some claim Leonardo da Vinci may have made it. Others argue it was the work of an unknown master, deliberately crafted to deceive pilgrims yearning for miracles. For more than 600 years, the debate has raged. Faith on one side, reason on the other, and neither has ever backed down. But in the 21st century, the era of pure philosophy and speculation came to an end. Science entered the scene, cold, precise, and uncompromising. Genetics, high-energy physics, spectroscopy, forensics. Technology made possible what was once unimaginable. Researchers stopped seeing the shroud as a sacred icon. They examined it as a crime scene, as evidence in an unsolved case, as a biological hard drive silently recording 2,000 years of history. They extracted and sequenced the DNA embedded in the fibers. They scanned every single thread with X-rays. They broke the bloodstains down into molecules, atoms, and elemental signatures. They expected simple answers, traces of a single individual, or perhaps remnants of pigment left by a medieval artist. But what they found was far from simple. The genetic analyzers at the University of Padua and the physicists working in the sealed laboratories of ENIA discovered results that left even the most experienced scientists shaken. The data didn't just question existing theories, it shattered them completely. Both skeptical explanations and traditional religious interpretations collapsed under the weight of the results. The shroud was no longer just fabric. It became a map tracing a journey that began 2,000 years ago, a record of suffering so precise it cannot be reproduced, and evidence of an intense burst of energy, one whose nature modern physics still cannot explain. This marked the beginning of the most extensive forensic investigation ever undertaken. We will follow trails invisible to the human eye, microscopic pollen grains, molecular residues, biological fingerprints, and step by step, we will uncover whose blood truly soaked these threads and why it seems to scream of torture spoken not in words, but in the language of biochemistry. We will examine the physics behind the image itself, an image that functions as a photographic negative, a hologram, and an X-ray all at once. We will also answer a question that stunned the scientific world: why DNA analysis forced researchers to admit that this artifact could not have been created in Europe, and that it bears witness to events that altered human history. To truly understand the shock of modern genetic findings, we must return to the very beginning of scientific shroud research, modern syndenology, back to the moment when humanity first truly saw the face hidden within the shadows of the linen. That moment came on May 28, 1898, in Turin. A lawyer, city council member, and passionate amateur photographer named Secondo Pia was granted rare permission by King Umberto I to photograph the relic during its public exhibition held in honor of the prince's wedding. At the time, photography was far from simple. It was slow, technical, and demanding. Pia hauled a camera the size of a suitcase onto tall scaffolding inside the cathedral. He relied on powerful magnesium flashes and lamps to cut through the dim interior. He exposed 2 large glass plates, each measuring 50 by 60 centimeters. Late that night, alone in his home darkroom, illuminated only by the faint glow of a red lantern, Pia lowered one of the plates into a bath of developing chemicals. And then it happened. As the image began to emerge on the glass, Pia nearly dropped the priceless negative in shock. What he saw felt unreal, almost supernatural. On the photographic negative, where light should turn dark and dark should turn light, there appeared not a faint, blurry stain like the one visible on the cloth itself, but a sharp, high-contrast, astonishingly detailed, positive image. A face, deep-set closed eyes, a broken nose, a mustache and forked beard, bruising on the right cheek. For the first time, the figure hidden in the linen was no longer a shadow. It was a man. The face carried an expression that was majestic, calm, and commanding, yet it belonged to a man who had just endured unimaginable physical suffering. The impact was overwhelming. It wasn't just a surprise. It was a revolution in how people understood the shroud. A normal drawing, whether made with paint, charcoal, or even blood, behaves predictably when turned into a photographic negative. Light areas become dark. Dark areas become light. The result is distorted and unnatural. The face turns into a flat, mask-like image. But the shroud was different. When photographed in negative, it looked like a true black-and-white photograph of a real, living person. That single fact changed everything. And to this day, it remains impossible to dismiss, no matter how skeptical or determined to debunk it someone may be. What we see on the cloth with the naked eye is not a normal image at all. It is already a negative, and that raises an impossible question. Who in the Middle Ages, in the 10th or 11th century, understood the principles of photography? Who knew that light could be reversed to create a hidden image? The answer is simple. No one who could deliberately create a perfect negative image without any way to preview or verify the result. Photography would not exist for another 800 years. The human eye and brain are not biologically capable of perceiving the world in negative, let alone reproducing it with such precise gradations of light and shadow. This was the first true mystery, the first crack in the seemingly solid wall of skepticism. The shroud did not behave like a painting. It behaved like a photographic plate, capturing a single moment in time. Over the years, the cloth has been examined with X-rays, ultraviolet light, infrared imaging, and laser scanning. Yet the greatest secret was never in the image itself. It was in the dirt, in the dust, in the microscopic particles trapped between the linen fibers during 2,000 years of movement, handling, and exposure. In 2015, a group of geneticists and biologists led by Professor Gianni Barcaccia from the University of Padua received rare permission to study the relic. Their goal was ambitious and unsettling at once: to find and examine DNA. They were not looking for the DNA of God. Science has no idea what such evidence would resemble. There is no control sample, no baseline for comparison. The researchers were not trying to identify one single person. They aimed to rebuild the story of the cloth itself, where it traveled, who touched it, and whose hands carried it through the centuries. To achieve this, they used specially created sterile micro-vacuum devices equipped with ultrafine filters. These instruments gently captured microscopic dust, pollen, and organic fragments, not just from the fabric surface, but from the deep gaps between warp and weft threads where ancient traces can stay sealed for thousands of years. They also studied particles collected during the 1988 restoration, which had been carefully stored in archival conditions. This effort stretched the boundaries of what science could safely attempt. Even the tiniest modern contamination, a sneeze, a breath, a single skin flake from a priest, conservator, or technician, could destroy ancient signals and replace them with modern genetic interference. The samples were moved into an ultra-clean, room-grade laboratory. There, sequencing began using next-generation sequencing, known as NGS. The scientists concentrated on mitochondrial DNA from plants and humans, and for an important reason. Unlike nuclear DNA, mitochondrial DNA appears in hundreds of copies per cell, is inherited only through the maternal line, and survives much longer in ancient material. It functions as a strong and dependable marker of geographic origin and human movement. For weeks, computers operated non-stop, decoding millions of nucleotide sequences and matching them with global genomic databases representing populations worldwide. When the final diagrams appeared on screen, charts of genetic haplogroups and ancestral lineages, the researchers understood they were seeing something that defied simple explanation or forgery. This was not one person's genetic profile. It was a genetic image of humanity itself. The results were published in the highly respected journal Nature Scientific Reports, and the response was explosive. The findings sent shock waves through the scientific world. The scientists expected one clear, dominant genetic signature. If the shroud were a medieval forgery made in France, as skeptics have argued for years, then European DNA, especially French or Italian, should have clearly dominated the results. If instead it were an authentic relic from Jerusalem that never left the region, then the genetic traces should have pointed almost entirely to the Middle East. That was not what appeared. What emerged instead was chaos. The entire world recorded on a single cloth. The shroud revealed genetic fingerprints from people across vast areas of Eurasia and Africa. First, the Middle East. Scientists detected haplogroups commonly linked to the Druze, a closely connected, historically isolated ethno-religious group living in mountainous regions of Israel, Jordan, Lebanon, and Syria. Druze DNA is considered extremely ancient and remarkably stable, having changed very little over thousands of years because of centuries of isolation. This makes it a strong marker of geographic origin and offers compelling evidence of a Middle Eastern link. Second, Western Europe. Haplogroups like U5b and H1 through H3 appeared exactly as anticipated since the 15th century. The shroud has remained in Europe, first in Chambéry and later in Turin. It was handled by poor Clares nuns who restored it, by members of the House of Savoy who possessed it, and by countless European pilgrims throughout the centuries. Third, North and East Africa. Haplogroup L3 and M1A1 appeared, pointing toward regions including Egypt and Ethiopia. This was unexpected and deeply fascinating, suggesting contact with some of the world's earliest Christian communities in Africa. Fourth, South Asia. Haplogroups M39, M56, and R8 appeared, genetic markers typical of the Indian subcontinent. And most astonishing of all were traces from East Asia. Haplogroups such as D4 and G2A, markers linked with China, were present. China, India, Africa, Europe, the Middle East, all encoded into one ancient cloth. How could this be possible if the shroud were a forgery created in the damp cellar of a French abbey around the year 1350? How could traces of Chinese and Indian DNA possibly exist on it? In the medieval world, globalization did not exist. There were long-distance connections, yes, but not on a scale large enough to leave such widespread, clearly identifiable genetic traces on a single piece of cloth. The answer proved even more extraordinary than the mystery itself. The shroud is not simply a burial cloth, it is a traveler. When scientists in Padua mapped the genetic data, they made a remarkable discovery. The genetic spread aligned almost perfectly, with precise geographic accuracy. With an ancient historical root many scholars had long dismissed as legend, the journey of the man-made image called the Mandylion. According to early Byzantine, Syrian, and Arabic sources, the shroud was folded in four, so only the face remained visible, displayed inside a special frame. This folded relic, known as the tetradiplon, did not stay in Europe. It moved throughout the ancient world. The journey began in Jerusalem, the site of death and resurrection. From there, it traveled to Edessa in the 2nd century. The shroud stayed there for centuries, hidden within city walls before rediscovery. Edessa stood at a crucial crossroads of the ancient world, the Great Silk Road. Caravans from China, India, Persia, and Arabia passed through. Merchants carrying silk and spices, pilgrims, diplomats, all came to venerate the relic believed to protect the city. They breathed near it. They kissed its casing. They touched the reliquary. With each encounter, microscopic traces, skin cells, hair fragments, sweat settled onto the cloth, layer by layer, century after century. The DNA of the world accumulated on its surface like unseen dust. The journey continued to Constantinople. From 944 to 1204, the relic rested in the heart of the Byzantine Empire. Byzantine emperors acquired it by purchase or force and brought it to what was then the world's capital. Constantinople was a true ancient megacity where people of every race, culture, and region gathered. Then, in 1204, amid the chaos of the Fourth Crusade, the city was violently looted. Afterward, the shroud disappeared. Next came Athens and Greece around 1205. Following Constantinople's fall, the relic passed into the hands of French knights and remained in Athens for a time, then France around 1353. This marks the shroud's first clearly documented appearance in Western Europe, held by the knight Geoffroy de Charny. Here, the genetic evidence becomes devastating for the medieval forgery theory. A forger working in 15th century Europe could never have collected dust and DNA from China, India, the Middle East, and Africa, lands known only through distant travel stories like Marco Polo's. Nor could he have intentionally planted those traces on a cloth to fool genetic scientists 600 years later. The DNA found on the shroud is not random contamination. It is humanity's collective biological memory. It is dust from the sandals, robes, and hands of countless people who gazed upon this face across thousands of years. This biological record proves the object's antiquity and Eastern origin more convincingly than any radiocarbon test ever could. But human DNA tells only half the story. The other half is written in plants. Palynology, the study of pollen, provided another critical piece of the puzzle. Two respected experts approached the mystery independently. Professor Avinoam Danin, a well-known Israeli botanist from the Hebrew University of Jerusalem, and Swiss forensic scientist Max Frey, a pioneer of forensic pollen analysis. Working separately, They studied pollen grains trapped deep inside the linen's fibers. Using adhesive tape, they carefully lifted microscopic samples from the cloth. What they uncovered caused a sensation within the botanical community. Pollen from 58 different plant species was identified on the shroud. 17 of those species are native to Europe, exactly what would be expected given the shroud's documented history over the last 6 centuries. But most came from elsewhere, plants native to the Middle East, Turkey, and the Anatolian steppe, perfectly matching the ancient route through Edessa and Constantinople. Then came the most remarkable discovery. A group of plants that grow nowhere else on Earth, species found only within a narrow geographic corridor between Jerusalem and Jericho. At the center of this finding was one plant in particular, Gundelia tourneforti, a thorny desert shrub, a type of thistle. Its pollen made up nearly half of all samples discovered on the cloth, an enormous and highly unusual concentration. Why would pollen from a thorny plant be so dominant? Why would thorns be linked to the burial cloth of a man treated as noble? The answer appeared when historians and theologians revisited descriptions of Christ's passion, the crown of thorns. Gundelia tourneforti, with its long, rigid, needle-like thorns, is exactly the type of plant Roman soldiers could have used to twist a mocking crown for the king of the Jews. Suddenly, the pollen began to speak. This plant blooms near Jerusalem at a very precise time, early spring, during the Jewish festival of Passover, Pesach. The discovery of an unusually high concentration of pollen from this exact thistle, clustered around the head and shoulder areas of the cloth, serves as direct evidence that the body was crowned with it. A second botanical clue came from Zygophyllum dumosum. This plant is endemic to a very limited region, growing only in the Judean desert and parts of the Sinai Peninsula. Its pollen was also found on the fabric in significant quantities. No medieval forger working in France could have obtained pollen from plants native solely to Israel and then applied it invisibly at a microscopic scale onto a piece of cloth. A forger would have used paint. Pollen cannot be painted. It behaves like an invisible seal from a crime scene, a form of geographic fingerprint that cannot be forged. For many years, especially during the highly rational 19th century, skeptics claimed the reddish marks on the shroud were nothing more than pigments, ochre, cinnabar, or tempera mixed with gelatin. That claim collapsed in 2017. A team of Italian researchers led by Professor Giulio Fanti of the University of Padua, working alongside physicians from a hospital in Trieste, studied the stains using transmission electron microscopy and Raman spectroscopy. By examining the material at the nanoscale, they saw not pigment, but blood, human blood type AB. It is one of the rarest blood groups, yet it appears frequently on ancient Christian relics, including the Sudarium of Oviedo. But this was not the blood of a healthy person. Inside it, scientists identified nanoparticles of creatinine and ferritin bound to hemoglobin. Such extreme concentrations appear under only one condition: severe fatal trauma, prolonged torture, combined with dehydration and massive muscle damage. When muscle tissue is destroyed by repeated blows, a process called rhabdomyolysis, creatinine floods the bloodstream in enormous amounts, and the blood on the cloth recorded exactly that.